Hello friends, today we are going to learn about the design of fermenters. We'll be doing this in two parts. The first part of the video deals with fermenters which are used to grow the aerobic organisms. Now fermenters are synonymous with this word bioreactor. So bioreactor or a fermenter is any culture vessel that is used to grow microorganisms under controlled conditions. Now, since we have to control the physical and the chemical conditions for growth, these fermenters are usually very expensive. Now, there are two kinds of cultures, two kinds of bacteria when we are trying to use them in the industry. One kind of bacteria which require a lot of oxygen for the growth, and the other types which don't need to be stirred and shaken, and their oxygen requirements aren't that high. So cultures can be standing cultures or aerated cultures, and most aerated cultures require a large amount of oxygen. The stirring of this culture not only provides the oxygen to be mixed well, it also mixes all the nutrients of the culture medium very well, makes it available for the organisms, and also helps the dilution of the waste products that are released by the microbe. So most of these aerobic organisms, as I said, are grown in what is called as stir tank reactors. The simplest form of stir tank reactor would be your Erlenmeyer flask, which is about, the carrying capacity is about 200 ml. And a more controlled variety of this would be a 1 liter table tank fermenter, glass fermenter. And a higher version of this would be a 1000 liter fermenter that is usually used in the wine industry. Now, how do these stir tank fermenters look like? This is an example of a very small stir tank fermenter, or tabletop one, which the capacity is about 1 liter. It's made up of glass and it has several attachments to it. The first part is the head plate or the lid of the fermenter, which has on it loaded a pump, an agitator pump. As the name suggests, the pump actually helps the agitator rod to move, and at the base of the rod you have the impeller blades, and these impeller blades actually carry out the mixing motion. The impeller blades look something similar to your blades of the fan, and as I said, the function is to mix the ingredients of the culture medium well. The top of the fermenter usually has a lot of gauges for checking out the air pressure. It has a lot of other things attached, such as the condenser to take care of the volume of the medium so that it doesn't lower because of evaporation. This is the impeller blade that I was talking of. There are a lot of ports attached to the fermenter vessel, usually at the sides, the bottom part of the fermenter. And these ports are usually sample ports which help one to remove the sample to check for the amount of the end product, the pH, and the concentration of a certain ingredient that is there available at a certain point, and most importantly to look for contamination. The bottom of the tank is also used to retrieve the culture medium after the fermentation is over. There is also a weighing cell that is placed at the bottom of the tank. The fermenters are usually double walled or jacketed to take care of the cooling and temperature requirements of the fermentation, and there is a control panel which actually automates the entire process of probing for oxygen, pH, etc., and makes it very, very user friendly. We learn about the other fermenters in the video too. Thank you.